building on the foundational questions and the promise and unique requirements of NK cells, I want to shift our focus to innovative technologies, platforms, and strategies currently setting the pace for NK cell therapies. So, Raphael, I'd like to talk to you again a little bit how you and your team at NKILT have developed your proprietary fit-for-purpose NK cell activation platform. Talk to us a little bit about how the concept emerged and what differentiates it from other NK or CAR platforms.
Yes, thank you. So obviously, when we decided to go with NK cells, I will say first that our technology, which we call chimeric ILT receptor, which is intimately specific to the target that we're looking after, which is HLA-G, and that has a unique property, which is we're not using an antibody binding, but we're using a receptor binding approach, which is kind of like it's the similar thing, but in our case, the importance of using a receptor instead of the antibody binding approach is that target HLA-G, which is expressed across all cancers in over half of cancer cells, is unfortunately expressed in multiple isoforms, which make an antibody likely to face difficulties down the road because it will be specific to isoform A. And then when the cancer cells is going to start to secrete isoform B and C, your antibody may have difficulties recognizing it because it is structurally different. So passing this, there's a uniqueness aspect of our construct there. But because we were going with NK cells as a first approach, we also did T cells, by the way, and they work very well also, but we wanted to have a allogenic approach, which is easier with NK cells. The first question everybody was throwing at us is that NK cells die out in 2 weeks. You need to find a way to activate them longer, because if you don't, it's not going to be strong enough or potent enough to kill cancer cells. So my co-founder and chief scientific officer, Henry Bell, actually had in the past developed a technology with NK cells that was leveraging a pathway called MyD88, and that is allowing secretion of IL-15, IL-18 that are two cytokines very important for NK cells to maintain activation. And there are multiple ways of doing this. He actually patented this in the previous life. And he used the same concept, not of course the same idea, because it has to be different and specific to what we're doing. But the same idea of saying, if we find a way to have our cells feeding themselves for long enough through the activation that we generate, it will allow them to maintain that serial killing for more than 2 weeks. And so we screened over 90 different potential activation domains that we will integrate into our construct. And we nailed down to a dozen and then down to 2 or 3 that were the ones showing not only the best killing outcome, so the highest potency, if you can say it that way, but also the one that will also allow the cells to grow the best. Because you need your cells to not only kill, but they need to expand, and they need to be able to grow in vivo and to continue to expand and reach out to the tumors. So at the end of the day, we have a handful of activation domain that we consider are the ones that are the most efficient and that are triggering the highest outcome. And we selected those and started testing them preclinically and also in vivo in the mouse model. And this is exactly where we are now. We are now launching extensive testing in vivo in leukemia, but also in several solid tumors, as we have shown multiple proof of concept in vitro across 6 or 7 different solid tumors, from bladder, pancreatic, esophageal, colorectal, and a few others.
Great, thank you, Raphael. And Tracy, I would like to offer you the same opportunity to speak on NKore's approaches. As we were kind of even talking about this concept before we signed on today, that the whole is greater than the sum of its parts. And you are doing similar work to, of course, NKILT, but that the patient in the end is what's most important. So talk us through a little bit about how your approach complements or differs from traditional NK or T cell therapies, and a little bit about the specific tumors or clinical settings where your strategy, NKore strategy, holds the most promise for the patient.
Absolutely. This is a really exciting topic for me to speak on specifically because the activation process that we use, I feel, is really unique. We are taking osteoclast, a cell made from the bone, and using that alongside probiotic bacteria to supercharge these cells and really truly focus on reestablishing the immune system in a very high-powered way. So what we're seeing when we take this allogeneic approach and we infuse our cells, and again, we're doing this in human beings right now at very low doses. We're starting at 300 million cells, then the second dose about 21 to 28 days later is 600 million cells, and then another 21 to 28 days later, we're doing 1.2 billion cells. So considering the field currently and the hundreds of the billions and billions of cells that they're giving day after day, week after week, we are taking a much lower dose approach because of the cytotoxicity, the gamma interferon release, the overall killing ability of these cells are quite phenomenal when we've looked and compared them to other NK platforms. What we are seeing with our NKs is that they are truly adopting that aggressive behavior of the osteoclast, and they are being fed and activated and supercharged through this probiotic bacteria feeding type of approach. And what we have seen thus far in the patients whose data some of them we literally just finished evaluating. I'm actually in Boston right now, about to fly home in a few hours from the event BPI East here. That's a 20-year running cell therapy conference. And I presented this data for the first time, and it was quite exciting because we're truly seeing an orthogonal MOA showing improvement of overall potency, function, persistence of patients' NK cells. It's leading to this enhanced killing, survival in the tumor microenvironment, and reconstruction of the immune system. So what we believe is happening, and again, there's some more tests that we need to do in order to understand at what day our supercharged natural killer cells are actually leaving the patient's body. We're seeing these cells come in and not just target the cancer itself, but we're seeing it repair the immune system and the patient's own immune system adopt that aggressive behavior of the supercharged NK cells when they're first given to the patient. We have a patient right now that has been over 12 months since his last infusion, and whereas his NK cell count was only 1.5% NKs, where a healthy person, you see 5 to 15% NK cells. He's had 2 infusions in total. The first infusion was around 190 million cells. 7 months later, we gave him 350 million cells. And now 12 months after that second infusion, his NK cells are still at 21%, which is considered a very high donor. And not only that, but we are using a combinational therapy with him. But in the data that we have tested and the results that we've looked at, we're seeing exhaustion in the BTK inhibitor, for example, that he was initially on. And then we're bringing in the natural killer cells, and we're seeing a continuation of cell death happening. Not only are we seeing cell death happening as the natural killer cells go up in almost a 1-to-1 ratio, but we're also seeing B cells come down in his B cell malignancy at the same rate that the NK cells are going up, which is giving us a really good sense that these cells are not continuing, they're continuing to function long term. The persistence is there. And what we have in this same patient are very excited to see is that on his last CT scan, again, post this second infusion of 350 million cells that he got a year ago, we are watching the tumors in his lymph nodes once again shrink by 50%. So we're seeing long-term killing. So what we are hoping we're going to continue to see, not just in leukemia, but also in solid tumors like my daughter's, where we're seeing necrotic tissue in this tumor itself, where it didn't shrink in 5 years on conventional therapies. We believe that we have an approach that not only is going to help target both solid and liquid tumors, but we believe it's going to help reinstate that overall immune function that's going to help the patient stay in remission once that remission point is met. So where we are currently in time is not only do we see something truly remarkable happening once these cells take flight, we're watching them train and recondition the body's own immune system. And our hope is that with this 3-dose escalation protocol that we're currently doing with these patients, we're hoping that not only will that eradicate their disease, but it will keep them safe long-term. Now, we still have a lot of patients we have to study. We have more longitudinal evaluations that we need to do, a lot of tracking in the patient's blood, and of course, scanning that has to happen. We're going to find out really soon whether or not this 3-dose approach is going to be effective in many different types of diseases, because right now we just treated 2 days ago our second glioblastoma patient. We've treated, as of today, stage 4 breast cancer, my daughter's low-grade brain tumor, recurrent CLL leukemia that had a mutation that took his outcome from very good to very poor. This is the gentleman now that has gone from 20% cancer infiltration that had plateaued with the Mayo Clinic therapies that were being given to now 0.23% cancer left in his bone marrow. We believe we're getting to the source. So we're hopeful that through a monotherapy and a combinational therapy, we're going to see real promise in eradicating many forms of diseases.